Muscle repair following injury involves a well-coordinated immune response. Macrophages, which are cells of monocyte origin, are central in the clearance of damaged cells. In the process of regenerative inflammation, macrophages also secrete growth factors that promote tissue repair. Damage clearing and regenerative inflammation are characterized by the sequential phenotypic transitions of circulating monocytes to pro-inflammatory and then to repair macrophages. However, still little is known about the spatial arrangement and temporal progression of macrophages through all phases of tissue regeneration. Transcriptomics is an advanced research method that explores the entire set of RNA transcripts in a given cell or tissue, revealing gene expression patterns. In this study, the authors combined single-cell and spatially resolved transcriptomics, complemented by immunofluorescence validation, to unravel the events following monocyte infiltration of injured muscle. The study employed acute muscle injury and Duchenne muscular dystrophy mouse models and identified various macrophage subpopulations. Their temporal and spatial distribution in the injured tissue was mapped and predicted that these cells organize in distinct layers called regenerative inflammation zones, which surround injured fibers to support muscle repair. Immunofluorescence validated and visualized the ordered distribution of the distinct macrophage subpopulations around damaged and newly formed myofibers to support all phases of regeneration. A marker of growth factor-expressing macrophages called GPNMB was suggested to be an important player in regeneration, with ATF3 as a key transcription factor in GPNMB expression and growth factor-expressing macrophage regulation. A macrophage subset with extracellular matrix-depositing genes was identified as a subset associated with human muscular dystrophy. Intermittent glucocorticoid treatment disrupted the formation of the regenerative inflammation zones, highlighting the delicate balance required for effective regeneration in diseases like muscular dystrophy. This study links the organization of injured and regenerating muscle at different time points to various functional subsets of macrophages, reflecting the spatially and temporally specific architecture of muscle regeneration.